Okay. Okay, here we go. Well, I'm Marta. Hello, everyone, and I'm happy to introduce our first speaker of BrainHack Donostia, Dr. Verena Heise. She has a research fellowship at the Institute for Advanced Study in Germany, and she has a quite standard curriculum. She's a molecular biologist, neuroscientist, and epidemiologist with degrees from the University of Heidelberg in Germany and the University of Oxford in the UK. And nowadays, she's focused on meta-research to investigate how open science practices improve the reliability of research findings in biomedical sciences. So in particular, neuroimaging and Alzheimer's disease research. And she will be talking today about robust research. So Verena, the floor is yours, so feel free to start whenever you're ready.
Thank you so much, that's amazing. Fantastic introduction as well. So now everyone knows everything about me and I don't have to tell you anymore who I am. That's brilliant.
Yes.
Great, so I'll start sharing. I hope you can see this.
You can see it, yeah.
Fantastic, good. So if anyone wants to have a look at the slides while I go along, you can also download them from the OSF. The link is down here on my first slide, and I will also repeat it at the end. I can actually, hang on, I'll just stop sharing for a second, and then I'll post the link in the chat, because that's always slightly easier. Hang on. Let's do this. Then people don't have to try and find the link. Here you go. Okay, sorry. So I'll just start sharing again. Great, yes. So I'm going to do a brief introduction to sort of what I tend to call robust neuroimaging. And I'm actually really grateful to the organizers for having me here, and also for doing this in a very sort of open science focused way. I think it's great to see that there are so many talks at the BrainHack that really focus on openness, open neuroimaging and related matters, which I think is absolutely brilliant. And I think we're starting to see how many early career researchers around the world are actually getting more and more interested in this topic. And so I'm going to give a brief introduction to different sort of techniques and different papers that you might want to read. And I hope you're not going to feel totally overwhelmed afterwards. But let's just take it away. So my first slide is always just my disclosure slide, which I think makes a lot of sense to have, just to tell you who's paying me to do something. So I'm currently a researcher here at Hanse Wissenschaftskolleg back in Germany. Like Marta said, I spent a lot of time at the University of Oxford before that, which is where I started getting sort of interested in reproducibility and open research related issues. And I'm going to talk about my activities there in a bit as well. I also have a couple of sort of non-financial relationships. I work on a number of advisory boards, for example, with the British Neuroscience Association. And this is basically just to tell you that I am a very biased speaker. I'm going to try and convince you today that robust neuroimaging is something you should be doing, that we do have something that I would term a replication crisis in biomedical sciences. And so I just wanted to be very upfront about this. At least in my mind, there's no doubt that we need to change our ways, and not just the biomedical sciences. I think it's much more general, actually. So what is the problem that we're trying to solve here? And I'm going to start with just a bit of terminology, because people tend to have slightly different terminology. So I'm just going to try and make sure that you know what I talk about when I use the term reproducible, for example. So to me, a result is reproducible if it can be recreated by others when they use the same data and analysis pipeline. So this is a very sort of short or relatively simple version of reproducibility, or limited version is probably the better word. And then replicability is what we tend to be aiming for in our research. So this means that the same scientific conclusion can be reached using maybe an independent data set, maybe an independent analysis pipeline, or maybe both. So this is more what we're trying to aim for in all our research. We hope that our results are replicable across different sites, across different labs and countries, for example. So what we're trying to solve here, or what I'm trying to solve, is the replication crisis. And I realize that the crisis is also being termed reproducibility crisis by some, but to me, the term that I use is replication crisis because this is, I think, what it's mostly about. It's the fact that we can't recreate results using independent data sets and independent analysis pipelines. And this is illustrated very nicely in a number of different papers, such as this, where they did a large-scale replication project in psychology, and they managed to, so that I think they looked at 100 different experiments that they found in the literature. They tried to replicate them, and they only managed to replicate between a third to a half of the original findings. And that has set off a number of different reproducibility projects. For example, we also have this one here in neuroimaging, where people try to recreate or see how replicable 17 different effects were. And we also see this in cancer research, where people try to do a replication project. And again, the sort of rate of replication was relatively low. There are other issues around this cancer biology project that are very interesting. For example, especially the issue that when we look at preclinical research, there tends to be a lot of information missing from the methods section of papers, so that it makes it even harder to replicate papers, but that's maybe a topic for a different conversation. There are also papers such as this that look at reproducibility in machine learning models, which is a huge topic of discussion at the moment. But I want to make sure that you don't think this is just to do with biomedical sciences. We know that, for example, in social sciences from this paper, that there are issues as well. So they looked at social sciences experiments that were published in Nature and Science and only managed to replicate around 60% of the findings. And so the issue that we're facing at the moment is that we have a huge amount of literature. And by that, I mean, you know, it's basically impossible to keep track of the literature that's being published in your own research field. And that's, but the research results that we produce don't seem to be reliable. And I think that's partly because we tend to get jobs based on the quantity of our publications and based on where we published them. So the outlet, the journal, and the impact factor of the journal, or, you know, standing in the field that this particular journal has. But we're not getting jobs based on whether what we actually found in our research was true. I mean, it's difficult to talk about truth here, but the point is that if we have a reliable results, we are much more likely to have something, to have found a true effect and not just a false positive effect. So I think what we're looking at in the literature is a huge number of false positive effects, unfortunately, and we have to find ways of dealing with this. Before I'm going to talk about ways in which we can deal with this, I'm going to give you a brief introduction as to why I care about this issue and why I think we should all care about this. So first of all, like Marta said, I'm a molecular biologist, neuroscientist, and now epidemiologist. So I do Alzheimer's research, and I care very much about my research results being useful in the clinic somehow, sometime down the line. You know, not maybe today or tomorrow, but maybe in a couple of years' time. And so when we look at papers like this, which was published by a pharma company, I think it was Bayer, again, they tried to replicate findings from the mostly academic literature, and they only managed to replicate a third of the findings. And yes, I realize that people are now talking about issues with this particular study, but I think it points to a larger problem, which is the fact that if pharma companies are supposed to produce drugs based on the research that we do in academia, then the results we produce need to be reliable. And so I care about this because I care about translation into drugs. It also has real life consequences, what we do. And this is sort of maybe slightly more recent stuff that's happened. I guess you will be familiar with these two papers that after a huge scandal had to be retracted, probably because the data that were used in these two papers are probably fake data sets. And so I think we have to be honest about the fact that what we do has or can have real life consequences, especially during a pandemic such as this one. So the consequence of those papers being published was that the WHO halted clinical trials because of safety fears that came from the conclusions of one of those papers that I just showed you. Afterwards, the papers were retracted and the WHO started the trial again. But I think we have to be mindful of real life consequences of the research that we do and the way that research results are being used. And I think this leads us to ethical issues. On the one hand, if clinical trials are started based on unsound preclinical evidence, then this, in the worst case scenario, might put patients at risk. For those of us who use animals in our research, I think we always have to be mindful that we're sacrificing them in the end. So I think we have a duty to get the evidence as right or as reliable as we possibly can. And I think on a more mundane level, it's also incredibly much research funding that gets wasted if we don't produce reliable results. And some people say it's up to 85% of research funding. I think the particular figure doesn't really matter. Even if it's just 50%, you know that this is going to be billions of dollars across the world, billions of euros or pounds, whichever currency you like, that is being wasted on unreliable research. And I think this also leads to issues such as trust in evidence. If I don't trust my own results, if I don't trust my colleagues' results, then why do we expect the public to trust, to believe in evidence? And I think that it's very clear in the current pandemic why this is a huge issue. We have politicians such as this one who say, we don't really care about what the evidence says. Or also, I think on a more sort of nuanced level, the evidence is out there. There are papers out there that say, there are specific links, but those papers are just, bad research, I would term them, and they are not reliable. But this is something that the sort of general public can't really tell. So I think we as researchers have to get the science right in the first place, so that we know that the public can trust what we publish, that we have politicians who can't sort of take our results and turn them into something that sort of helps their own agenda. And I think even if you think this is way too high level for me, there are really, really good reasons, selfish reasons, why you want to work reproducibly. And Florian has this great paper out, and I really recommend this to you, which basically says, well, there's no way that you can send an email to your past self from 6 months ago or 6 years ago. I think many of us will be familiar with how it goes with publishing. So you send something out for review, sometimes you get the reviews back a couple of months later. The reviewers ask you to do something, some additional analyses or redo some analyses you've already done. And then unless you have code that actually allows you to understand how you analyze the data, it will be very, very difficult for you to do this. Sometimes we start new projects in the meantime, and it will be very hard for you to really try and understand, how did I do this again? And there is no way that you can email yourself. You can potentially email other authors of your paper, but in the end, you're the one who did the work, so you should be able to sort of work reproducibly just for selfish reasons to get the science as right as you possibly can. And so I'm going to talk about a couple of solutions here. And yes, part of the solution, I think, lies in open science, and especially those parts of open science, so making data available, using open source software so that other people can actually redo the analysis you did. If you're using open source software, they can just download the software that you used. Open methods and open access publishing. I think one of the items that's really missing on this slide here is open research culture. That's something that I feel very strongly about. I think we need to worry very much about diversity and inclusivity in this space. So openness also means who we are open to. But I'm not going to talk about this at all. I saw that Cassandra Gould-van-Praag is one of the speakers later this afternoon, and I think she's going to do a great job of explaining this in much more detail to you. And when we look at open science in the current pandemic, I think this is one of the very few positive things that have come out of this. So there is a statement on the Wellcome Trust website that a lot of publishers, for example, have signed up to. So they basically said, we're going to publish all COVID-related papers with open access. There's a lot of open data and open materials happening. There's a lot of sharing happening at the moment. So I really hope that this is going to outlast the pandemic, and this is going to change the way we do research in a much more fundamental way. But I think open science is only part of the process. The rest, or the sort of missing half of this, is really good research practice and going back to first principles and thinking about what does actually good research practice look like. And this is something that as early career researchers, we don't really get taught very much. What is the process of doing research? And I think, marrying this up with the open science infrastructure and open science ideas, I think gives us the ingredients that I tend to call sort of robust research. So I'm going to go through the cycle of a research project in the next couple of minutes and think about at each of those individual stages, what does good research practice look like, and how can we ensure that we're doing the best we can at each of these stages. And the first stage, I guess, is asking the right question. So how do you get the right question? And that's obviously a very difficult question to answer, especially you know, as a student, you might just work on questions that your supervisor came up with. But in later stages, you're going to develop your own questions. And where do they come from? Well, I think the first thing that we should all be doing is actually doing much more systematic literature reviews. This is something that sort of in my previous life as a molecular biologist, but also in neuroimaging, I didn't really do very much. But as an epidemiologist, you're starting to realize how important it is to do systematic reviews and to understand where exactly are the gaps in the literature. So this is not just about picking the 1 or 2 papers that sort of fit with your hypotheses or that you really like. It's about really looking at the entirety of the literature to try and understand, you know, what have people done before, where are the gaps, what are the things that are still outstanding, the questions that we should be looking at. And there are some great sort of ideas for how to do this in neuroimaging. So there's this paper here that looks at 10 simple rules for neuroimaging meta-analysis. And there is this tool here, neurostrength.org, which I really recommend to you. So this is a way for doing meta-analyses or looking at meta-analyses, especially for fMRI studies. And so the other point that I'd like to make here is that asking the right question is also about interacting with people. So speaking to colleagues, speaking to clinicians if you're working on a clinical question, but also involving the public and patients. And I think this can be very powerful because the questions that patients have or that clinicians have can be totally different from what you know, sitting at your desk are actually thinking they might have. So I think there are great ways nowadays to interact with others, interact with the public via social media, also via citizen science projects, and just trying to understand, you know, what do taxpayers actually want us to do with their money that they give us, I think is a very important thing to consider. And so for me, this is about being open and collaborating already at the stage when you're just developing your research question. Right, next stage, study design. If you ask me, I think study design is where most of this goes wrong. And I think study design is something that we should talk much more about and should be much more careful about. Because if you don't get the design right, then there's very little you can do at this sort of analysis stage later on to sort of save your project, if that makes sense. So I think the first question really needs to be, do you need to collect new data? There are so many data sets out there, especially when you're doing MRI research, for example, that you would never be able to collect in the space of your own PhD project or, you know, your postdoc that you have for a year or 2 or 3 if you're lucky. So I haven't personally tried Google Dataset Search. I'm not sure if it's a good tool, but it's just a placeholder for me to tell you that there are basically data sets out there, such as the UK Biobank. So this is a data set that I work with and I find absolutely brilliant. So they have now scanned 45,000 participants. So they have MRI data, brain MRI, and also heart MRI and some other imaging modalities of 45,000 people. And this is something, you know, when you think about it, how many people are you going to study within your PhD? How many people can you scan on one single MRI scanner? You know, if you're lucky, maybe 100 or maybe 200, but not 45,000. So maybe think about what kind of questions you can answer with these types of data sets. But there are lots of other data sets out there. You can look at the Connectome data sets. You can look at the International Neuroimaging Data Sharing Initiative to try and find out what other data sets are out there that you might be able to use to answer the question that you're interested in. Secondly, if you then decide that you do need to acquire your own data set, then I think it would be really good practice to look at reporting guidelines. And the Equator Network here is a great tool to look for reporting guidelines in different fields. Here are the ones that are used in neuroimaging. And I think it's very powerful to be able to look at this and try and understand what you're supposed to write about in your paper afterwards, because that gives you an indication of what you need to look out for in your study design. And the study design is basically about 3 major questions, right? So the first question is, how do you deal with bias? And when we look at clinical trials, the way we deal with bias is by blinding people, because, you know, the ways that bias can creep into your project, for example, is if you as a researcher know whether a patient is in the drug group or in the placebo group in a clinical trial, then you might be asking patients slightly different questions. Or the other way around, if a patient knows that they are in a certain group, they might be reporting side effects differently depending on which group they are in. So this is how bias can creep in, and this is where blinding helps a lot. The other issue that we need to worry about is confounding, and the sort of most commonly used confounders are things like sex and age, but there might be others that are important for your own study, and you might want to think about how your study design can tackle those confounders and ensure that they are spread equally across the groups, for example, if you're looking at 2 different groups. And then the last question is, how do you deal with chance? So how do you reduce the chance of getting a false positive or a false negative result? And this is where we need to talk about sample size and statistical power, at least for those of us who do null hypothesis significance testing, which I assume is still most of us. I'm not a statistician. I don't know very much about Bayesian statistics, but to me it sounds like a very great way to do things differently. But for those of us who do null hypothesis significance testing, we need to worry about statistical power and sample size. And there is this paper that I just showed you that basically says our sample sizes are too small, and this undermines the reliability of neuroscience. And it basically says the median statistical power is 20%. In animal studies, it can be slightly higher, but in neuroimaging, and the paper is a couple of years old by now, so I think it's getting better in neuroimaging. But in their paper, they say the mean statistical power is 8%. So why is that a problem? So the chance of finding a false negative is 1 minus power. So in this case, it would be 92% for neuroimaging studies. But even if you find a positive effect, a significant effect, as we would call it, the likelihood that this is actually clinical trials. So you might know about clinical trials that they have to go into registries before people even start collecting any data. You have to register, who exactly is going to go into your study, how you're going to analyze the data, and so on. And that's exactly the same here for pre-registration. So basically, you write up your hypothesis, you write up your study design, and your detailed analysis pipeline. So you basically write up your introduction and methods section of your paper before you start data collection. And then you can pre-register this online. There are a number of different registries nowadays out there. I personally use the Open Science Framework, but there are others. And the pre-registration will be published online. You can put an embargo on it. You can say, I want to embargo this for 2 years, for 5 years, however long you like. But the point is that others can ask you about your results. And I'll get to why this is important in a second. I think the important thing to say about pre-registration is that you can still do exploratory research. You can still explore your data to death and analyze the data in hundreds of different ways. But the point is that when you write your paper, you say, well, this was my original hypothesis. This is my pre-registered analysis. Here's what I found. And hang on, I actually did some exploratory stuff and found some really interesting stuff too. But the exploratory stuff is probably something you will want to follow up on in a next study that is then again pre-registered. You can find more information on this in this website. And so why am I a big fan of pre-registration? I think it gets around a number of different issues that we have and that are so nicely illustrated in this paper here by Markus Munafo and others. It gets around harking. So harking is hypothesizing after the results are known. This is basically the idea that people change the stories or change their hypotheses after they've seen the results to make the data fit the story, or the other way around, to make up a nice story around the data that they see. And this is a very common practice, but it's actually the opposite of what we should be doing. So we should state in advance what's our hypothesis and then interpret our results with respect to that hypothesis, rather than pretend that we looked at a completely different one. And then the other issue it gets around is also p-hacking. P-hacking is basically the problem that people tend to analyze data in hundreds of different ways, but they only report the one that gave them a significant result. And by definition, if you do null hypothesis significance testing, if you do 100 tests on the data, 5 of them are going to give you false positive results. That's our alpha of 0.05. So if you do hundreds of tests, then of course you're going to get significant results. And unless you report that you've done all those additional tests, then people who are readers of your paper won't be able to tell, did you get those results just by chance, or are they actually reliable because you did lots of tests and managed to get those sort of what you might want to call significant results by testing the data in lots of different ways. Pre-registration does not necessarily get around publication bias and low statistical power, but this is where registered reports come in. So this is a very new model of publishing papers or sending out your manuscripts. And the great thing here is, I think, that it doesn't matter whether you have negative results, null results, or positive results. Your paper is going to be assessed based on your question you're asking and on the study design. So, like I said before, you write up your introduction method section before you start collecting data. And at that stage, you send off your paper for peer review. And it's then going to be assessed based on the question you're asking and the study design, whether this is a paper that this particular journal wants to publish. And then you, if they say yes, then basically you get what's called in principle acceptance. And if you collect and analyze the data in the way that you described in your study design, then basically at the stage 2 peer review, the peer reviewers only check that you analyzed the data and collected it in the way that you prescribed. And then they say, okay, it's going to get published. It's not going to get published based on whether you have good results, results that fit your idea or hypothesis or whatever. It just gets published based on the question that you're asking and the study design. And I think this is a great way of getting publication bias. So publication bias is the issue that basically a lot of the literature that we read has significant or positive results. And that's because we have this impression that negative or null results are not as publishable as you like. And registered reports get around this issue because basically your study is going to be published completely irrespective of the results that we have. It also gets around the issue of low statistical power because registered reports have to come with a certain amount of statistical power. For some journals that's 80%, for some journals that's 90%. So this is a way to increase statistical power as well. So I'm a big fan of thinking about new sort of publishing models, and I think this is one of the best ones that we have now come up with. And why is pre-registration important, especially for MRI analysis? Well, you might know from this paper by Ross Poldrack that when you look at the bottom of this figure here, there are around 70,000 different ways in which we can potentially analyze neuroimaging data. And if you think about this, you know, if I just talked about p-hacking, where, you know, when you do 100 tests, you will get 5 false positive results just by definition. If you're thinking about 70,000 different ways of analyzing the data, you can imagine how many false positive results you will get just by definition of the method you're using. So pre-registration gets around this because then you pre-specify which analysis you're going to do, and you're not going to, you can potentially try all those 70,000 different ways, but then you pre-register this, and then people will know if only one of them gave you a significant result, then people will understand that, you know, this is potentially a false positive result. And there is also this great paper that was published only this year where they looked at the variability in the analysis of a single neuroimaging data set that was analyzed by lots of different teams. And again, this just showed how many different pathways we have to get to a result and how reliable or sometimes unreliable the results are that we get from these different analyses. Right, so once we have our study design and our pre-registration, I think what you also need to think about is how you're going to manage your data. And this is something, again, that you should think about at the study design stage. I'm a big fan of using the Open Science Framework, but this is just because I think it fits my purpose. Others might want to use completely different tools, but this is just an example of an easy way to sort of structure your data set. What you can see here in the components are different paths that you may or may not have in your own project, and this is a way to manage your data in a relatively easy way, and then make it publicly available later on. I'll talk about this in a second. And also, I guess this is something that you will talk about over the next couple of days as well as BIDS, so that's the brain imaging data structure. I think it's a great way to put your data in this particular structure, because then other researchers, it will be much easier for other researchers to be able to look at your data and understand exactly you know, what they're looking at. Are they looking at structural data, functional data, and so on? Right, that was study design stage. I spent a lot of time on this, partly because I think it's very important, and partly because I think this is where a lot of stuff goes wrong. So let's have a quick look at data collection and data analysis stage. First of all, again, I think the first question should be, do you actually need a new method? Do you need to develop a new method to acquire or analyze your data? There are lots of protocols out there. There are websites such as this one here, protocols.io, where you can try and understand how other people collected or analyzed their data, which might be similar to yours. There's also the NITRC that you can, again, look up to see how other people might want to analyze the types of data that you have. And I think when it comes to neuroimaging, I think we need to be much more careful about how reproducible our measures actually are. And that's about two main questions. So the first question is validity, and validity basically means, do you get the right answer? And then it's about reliability. So do you get the same answer twice? And just to illustrate what this means, I have this target practice here. So on the left, we have a result or a measure that's reliable because it's always in the same corner of the target, but it's not valid because it's not in the bullseye. Then next to it, we have a measure that's valid because when you take the average of all those measures, then you will end up in the bullseye, but it's clearly not reliable because it's all over the place on the target. And both of those measures are actually useful measures. So the reliable but not valid one is actually useful if you know what the correction factor is. So if you know how to get the measure, for example, if you have a scale that's always off by 2 kilograms, then you know you have to add 2 kilograms to all of the measures that you have. And this is basically a measure that's reliable but not valid. If you have something that's valid but not reliable, again, it can be useful. You just need to know that it's not reliable so that you take lots of different averages so that you end up in the bullseye in the end. What you don't want is something that's not valid and not reliable. And the case valid plus reliable is obviously the best case scenario, but it's actually quite unlikely that we have these types of measures. So first question is, how reliable and valid are your tests? Can you compare them with a gold standard or well-established tests if you develop new tests? And there is this very nice paper here that looks at reliability of neuroscience research, which I think is very useful to look at. Then the second question is, are you doing any quality control? And what types of quality control are you doing? So do you control your experimental setup, the reagents that you use, the data you acquire, the computer code you write? And I think this is all stuff that we aren't necessarily taught as much as we should be, but we all should be thinking much more about how do we do quality control. Then the question is, how do you get your analysis pipeline? Do you write your own computer code? And I guess like many others, I'm not a computer scientist at all. I've never been trained to write computer code, but still I do it to analyze my data. So the best way to do this is, on the one hand, use well-established pipelines and maybe grab codes from other people or people around you that are much better at coding than you are. And also, like I said before, using open source tools. I realize that MATLAB is something that's very often used still in neuroimaging research, but MATLAB has the problem that it's proprietary software, so other people who don't have the sort of financial means to use MATLAB might not be able to reproduce your code or reproduce your results. So I'm always a big fan of saying we should be using open source tools like R, like Python, and I'm sure there are others that might be useful for your own research. And then the last issue is following good programming practice, and I think you're going to hear much more about computational reproducibility in the next couple of days as well. So I always say there should be no miracles. I love this cartoon here. There should be no miracle in how you get from your data to your results. You should be able to explain this in a reproducible way and use tools such as electronic lab notebooks that allow you to have reproducible workflows. I personally use R Markdown in my own research. You might want to use Jupyter Notebooks, for example. So I think there are lots of different ways in which you can do this nowadays. And then the last thing on my sort of list of how reproducible are your measures is also the question, how good is your statistical practice? And related to that is the question whether you can actually embed a replication in your project. So you might know from genome-wide association studies that people who try to publish those types of studies now have to basically replicate their findings in 2 data sets. So they have the discovery data set, they find some specific single nucleotide polymorphisms that are different between 2 groups, for example, Alzheimer's patients and healthy participants. So you have to find those in a discovery data set and then validate them in a second independent data set. And I think this is a very interesting way of thinking about your research, whether this is something that you could potentially do in your own research as well. And then when it comes to good statistical practice, I think this is something that a lot of us are not trained in as well as we should be. And this is where we always should be looking to find people who are experts in statistics. And some of us are very lucky in that we have institutions or work in institutions that have statistical help desk, for example. So I would always recommend going to them, having a chat about your study design, even before you start to analyze your data, so that you can understand what's the best way to analyze the data that you're looking at. And I guess especially in neuroimaging, we always need to worry about this because of this study here, which also won the Ig Nobel Prize. I guess many of you will know about the sort of thinking salmon, or the salmon that, the dead salmon that actually manages to look at brains and have brain activity when the salmon looks at pictures of faces of humans. And so this is just showing you that we have huge multiple comparisons problems in neuroimaging, and there are ways to correct for this, and we have to use correction, because otherwise you will find brain activity in a dead salmon, which is quite unlikely to occur. So last stage, interpretation and publishing, and this is what I term open reporting. So what does open reporting look like? The point that I'm trying to make here is that on the one hand, you need to publish all the analyses you did. So you might have done some pre-registered analyses and you did some exploratory stuff as well. Publish all of it so that your readers know how exactly you explored your data as well. And obviously publish all the results, not just the significant positive ones, publish your negative results as well. Publish according to best practice guidelines, that's something I showed you earlier. But also, I think this is something we don't tend to talk about as much, is being honest about your biases and conflicts of interest. And many of us think of conflicts of interest like being something, well, like I'm getting paid by a pharma company to do something, and so maybe I'll interpret the data in a more favorable way because the pharma company is paying me to do this. And that's actually not what I'm talking about here. What I'm talking about here is the conflict of interest that all of us have with our next paper, that our next job is probably dependent on getting this paper out there. As students, as postdocs, we need to publish as much as we can, or we are told we need to publish as much as we can. And so our conflict of interest is always that we're trying to get this next paper out there. So let's just be honest about this, and be honest about the fact that we all have conflicts of interest and that we all have biases. We're all human. We have our favorite pet hypotheses. We're probably trying to prove those hypotheses rather than disprove them. And so this is a bias that we come with and that we bring to the data and the results that we're looking at. So the last thing that I want to talk about when it comes to open reporting is using preprints. For those of you who are not quite sure what preprints are, so this is basically the sort of final manuscript version. While you send this off to peer review at a specific journal, you can also post this on a preprint server. There are a number of preprint servers listed here. And I think this is a wonderful way for early career researchers to get your stuff out there, to get your study out there. And we all know, I said this before, how long it takes for peer review sometimes. It can take months until something gets accepted in a journal. And in the meantime, you might apply for a job or you might apply for a grant. And the great thing is about preprints is that you can put those preprints on your CV so that your future employer can read this, or the sort of grant funding reviewers can also read this. And I don't know what the situation is like in other countries, but in the UK, a lot of the funders have now said, they totally accept preprints as part of your publication list, and it doesn't all have to be peer-reviewed journals. What I'm not going to talk about is publishing open access, partly because this is something, at least in the UK, that we all have to do from 1st of January 2021. Most funding agencies in the UK now require you to publish with open access, so this is a done deal for me. It's a discussion we don't need to have anymore. I personally don't see any reason why we shouldn't publish with open access, but I'm very happy to have a chat about this afterwards. I realize not everyone feels as strongly about this as I do, but I think there is absolutely no way that we can get around publishing open access. I don't see why anyone would publish in any other way. And I personally don't care about the impact factor of a journal as much as whether they publish my paper with immediate open access, and also some other issues that I'm happy to talk about in the Q&A later. Then it's also about publishing your data and materials. And I would just encourage everyone to think carefully about whether you can actually publish your data sets. I realize that there are barriers to publishing data sets, especially when we're thinking about, for example, structural MRI. Your structural brain image is basically like a fingerprint. And I wouldn't want my fingerprint to be out there, even if it's anonymized, even if it's defaced. The structure of your brain is very unique. And so I would always encourage us to think about what are ethical ways in which we can make open data happen, but maybe not make it as open as just publishing it freely on a website. And obviously there are lots of different repositories for publishing data and materials, depending on which area you work in. I've showed you the Open Science Framework before, and the only reason why I'm talking about this so much is because they have this cute little button here in the top right-hand corner, which is absolutely brilliant. So you can make the parts of your data set, the parts of your methods that you want to make publicly available just with a click of a button. So if you use the Open Science Framework for your data management in the first place, then it's super easy to make this publicly available just with a button click, and that's the whole beauty of it. Also, if you're doing MRI analyses, there are great ways to publish unthresholded statistical maps on NeuroVault, and I totally recommend this if you publish any kind of MRI data. I think this is a super useful way of getting your unthresholded maps out there so that people can look at them and not just see, you know, what's below a relatively arbitrary threshold that you might have used for your publication. So in summary, robust research for me is about openness, open research, and then the other part is about good research practice and bringing those two together where it makes sense. And there is this great paper here that sort of looks at all sorts of different practices that I can also really recommend to you. So I realize I've talked about a lot of different practices, and this is exactly how you might be feeling right now. The nice thing is that we're recording this, so you can go back to the relevant parts of this talk whenever you like. And I really want to stress here that I don't expect everyone to do all the things that I talked about today. I think it's really important that you just pick the stuff that's useful for your own research. And I always use this tagline from a big UK supermarket, which is every little helps. And so if we just use one practice and then build on this over the next sort of couple of years, I think this would already be amazing. But I think it's also very important to note that this is not just about individual researchers. It is about the system. It is about the question, for example, what are the incentives and what are we supposed to do? So are we incentivized to make our research openly available or not? If we're not, then I don't think any of this is ever going to take off. So we have to talk to institutions, professional societies, funders, publishers, researchers. We all have to get together to think about how we're going to change the system to make it in some ways more efficient, but I think just you know, more open and more open to discovery, if that makes sense. This is where we need to get together, I think, and this is where I've done a lot of work of getting groups of people engaged. So this is where I'm going to get slightly political here, and I actually don't apologize for it. I think it's very important. So in Oxford, we set up something that's now termed Reproducible Research Oxford. We started this actually quite some time ago now, and this is just to show you our list of demands to show you that we're actually very nice people and friendly, and we love things like unicorns, rainbows, and puppies, not just open science. We started our initiative in 27. It was mainly early career researchers in the beginning. I think it's also very important to say that we have been supported by amazing PIs from the start and great collaborations with the library. So I think it's always very important to try and find collaborators within your university that can help you achieve what you're trying to do. And really the aims of Reproducible Research Oxford, at least to my mind, are improving training, improving infrastructure, incentivizing open and reproducible research, and really also creating a meta research community, because more of us are getting interested in doing meta research as their own research field. And I think creating a community around this can be very powerful as well. I think in the first instance, we have very much focused on doing training. And this is what you see here, seminar series, journal clubs, and so on. And I think this is for a simple reason, because training, all it takes is a crazy person like myself and a room, and then I need to find some people who want to learn more together with me on, for example, computational reproducibility. How do I do this? Improving my own skills and art, whatever you might want to think about. And so training has been sort of the first step for us, but we're now starting to think really much more about the bigger picture. What types of infrastructure do we need? What types of incentives do we need? And how can the institution help us there? Just one more word on the Journal Club. Reproducibility is something that started at Oxford. It's now present in lots of different institutions around the world. So I think this is always the lowest barrier, easiest way to learn more about reproducibility, especially if you work in a psychology-related field. And so try and find out if you have a reproducibility in your own institution, or maybe think about starting one, because I think this is a great way to start a community in your own field. So this is just a slide I leave in there, if you're, just in case you're interested, to think about what types of training are required. And if you might think that, well, you know, this is just a crazy bunch of people in Oxford who set up this network, well, what does this have to do with me? I would actually argue that there are lots more people around the world, especially in the UK, who are interested in driving this and driving change of the systemic change and cultural change in academia. And this is why a number of PIs got together to start the UK Reproducibility Network. And what we see now is that the Reproducible Research Oxford group is basically the local version of this network in Oxford. And we have lots of other local networks as well. And many of them are led by early career researchers. And I think it's just brilliant to see how many people are getting involved in this. And this is something that's not just happening in the UK. I think it's also happening in a number of different countries now. There are also some really great societies. And again, I'm super biased here because I work a lot with the British Neuroscience Association, but they're doing some really great work on credibility in neuroscience and improving the credibility and the trust that we have in research results. And also on a political level, there's quite a number of things happening. So the UNESCO, for example, is currently working on a set of recommendations on open science globally. So I think this is a super interesting initiative as well. So it's not just a couple of crazy researchers. There are even, you know, what I would call politicians nowadays that get interested in these questions. And I think if there's one good thing to take away from the current pandemic, it's this. It's the fact that open science actually was promoted a lot and has opened lots of different doors. And I really, really hope that this is something that will sort of outlive the pandemic and will change the academic community in a very fundamental way in the next couple of years. So in the end, I just want to say thank you to all the people that I work with in Oxford, in Berlin, and in other places. And thank you for your attention. Do get in touch if there's anything I can help with. And like I said, you can also just download my slides online. So now I think we're going to have a Q&A.